Jim, can you take now there's one chair up here. The university doesn't have any density limits these days, so it's whatever people are comfortable with. Okay, upcoming seminar announcements. IB seminar tomorrow at 12:30 is Gerald Carter from Ohio State speaking on formation of cooperative relationships in vampire bats. This looks like it's going to be really cool. If you look at his website, it's really interesting work. So I encourage everyone to attend that. You should attend every IB seminar. It seems like it's going to be very relevant for those of us interested in vertebrate biology. ESSIG brunch Friday at 10 is Hollis Woodard, assistant professor at UC Riverside, conservation physiology of bumblebees. On Monday, the IB seminar at 4, this is our last candidate for the herbarium directorship search, Tia Lynn Ashman from the University of Pittsburgh, understanding polyploid success across ecological scales. So there was really nice attendance at our last one with Lucia Lohman. Let's have similar attendance at our very last one, which is this, I'm sorry, Monday, the 28th. So that's a week from Monday. Herb group, also a week from Monday at 6:30 is Aaron Westing, our own Aaron Westing, and Jose Gabriel Martinez Fonseca, and no titles for those. Is Aaron there?
I'm here. Jose's going to talk about his experience surfing in Nicaragua over the past 2 decades, and we're also going to give a little bit of a perspective behind our new surf book that just came out.
Wonderful.
It's a really awesome surf
Congratulations. That's a week from Monday. Next week is spring break, just to remind you. Fossil Coffee on Zoom a week from Tuesday is Mackenzie, Kirchner Smith, Jameen Lee, and Tanar Frank, speaking on a variety of different things. Mackenzie, are you here? Okay, so that's Fossil Coffee a week from Tuesday. Next week, no seminar here because it's spring break. The following week is Mishi Tobler, a student invited speaker from Kansas State University. And David, or I think you're, or who's, okay.
There are still some spots on Mishi's schedule, so if you're interested.
Okay, any other announcements? Well, I'm delighted to see that somebody knows how to finish in style. Well, I will say congratulations to Michelle right at the outset here. I'm going to turn it over to Chris.
That was a great introduction already, Michael. So finally, you all know Michelle. That's what's so special here, right? She's confident, she's outspoken, she's passionate about science, statistics, outreach, mentorship, and you know, she's a total boss. She's a total leader, right? This is what we want to see in a scientist. Michelle started her career at University of Illinois as an undergrad, followed by kind of sort of intern work at the zoo, where she was doing both animal enrichment as well as animal behavior, starting an early on interest in animal behavior. Then she did a master's with Becky Fuller, also at the University of Illinois, on reinforcement in these cute little fish, which will be the theme today, the little rainwater and bluefin killies, where she looked at sort of pre-mating isolation. And then she joined my lab in 2017, where basically she was already fully formed. She was already a behavioral ecologist, and you could see it in the letters. And I mean, I want you to imagine like Athena being born from the head of Zeus, sword in hand. I mean, this is what I think of when I think of Michelle. Right? In 2 years, she managed to not only publish 2 papers from her master's degree, but then another, basically she could have finished in those 2 years, because she basically finished another 3 chapters. So 2 were published on our Pupfish system by 2019. I think a 3rd was already in draft form. So she really didn't have to move here. She could have graduated in 2019 and stayed in North Carolina. And over the course of those 2 years, she didn't just stick with behavioral ecology, right? As you'll see, she expanded first into aggression behavior and sort of agonistic behaviors. Then she expanded into kinematics, functional morphology, an entirely new field of behavior. And then for her final chapter, that's in revision now, she then expanded into quantitative genetics and QTL mapping of behavior, right? So Michelle, has expanded from this behavioral ecologist to sort of absorbing all aspects of behavior that you'll see today. But it's not just about the science, right? And even in her application letter, Michelle states clearly, right? Science means nothing if we don't communicate it. She's been a dedicated mentor and teacher and outreach. She's helped to lead Expanding Horizons within our lab, which helps to inspire young girls to go into science. She's mentored enormous teams of undergrads, some of who are here today, sort of day-to-day supervising, I think, at least 2 senior honors theses. As well as pretty unique is that every one of her chapters, the data has been predominantly collected by undergraduate authors. So Michelle would much rather sort of inspire a team to and teach the team how to collect data and then teach them how to analyze the data and ultimately analyze that data and turns it into papers, right? Just like a PI. Michelle's like, yeah. And as well as following the Black Lives Movement, she was one of the undergraduates here who led an effort to address a letter to the MBZ faculty about reforms that we should be doing. She joined many of the committees looking to increase inclusion, diversity, equity within the MBZ. She's also, in addition to developing curricula standards for the state of Illinois while she was a master's student, she went on to sort of leading her own groups when she taught with me, where she would sort of supervise whole teams of undergrads in doing research. And then for her final year here at the MBZ, she decided to co-teach, along with Emily Richards, her own course, meant to inspire undergrads to go into science. So entirely on her own, wrote it, taught it, she's teaching it now as well. I'm probably forgetting a whole bunch of things, right? There's so many outreach things that Michelle has done as well. But I'll turn it over to you, Michelle. And afterwards, you're all welcome to join us for champagne and cake, and I think we'll be in here. But with that, take it away.
Wow. Thank you both, Michael and Chris. Those were very touching and I really appreciate that. That's very nice. So hello, everyone. My name is Michelle St. John. I am a PhD candidate here at Berkeley, and I want to welcome and thank you all for coming to my finishing talk. So I am an evolutionary biologist, and I'm interested in understanding biological diversity. So I'm happy to say that I'm giving this talk here in Grinnell Library inside of the Museum of Vertebrate Zoology, and we have outstanding examples of diversity all around us and explicitly out in our collections. But I am a fish biologist, so I'd like to give you some examples of fish diversity to get going. So something you might know about or not know about me is I actually collect fish illustrations. And so these are illustrations that I've gotten from like thrift stores and garage sales. And I scanned these all onto this PowerPoint for you all today. But even briefly looking at this, we can see many different types of diversity. So there are examples of morphological diversity. There are examples of dietary diversity, diversity in parenting behaviors, and diversity in behavior in general. So as an evolutionary biologist, I'm not only in awe of this diversity, but I'm also curious about how it comes to be. And my approach to this during my PhD has to understand diversity through the lens of evolutionary novelty. And before we go on, I just want to point out my favorite fish out of this is this very grumpy goldfish here up at the top. I don't know who drew that, but I just absolutely love it. Okay, I'm going to shamelessly steal this quote from a paper that was published last year, and it says that understanding how biological diversity comes to be requires appreciation of evolutionary novelty, and I'd like to tell you why. So first, what is evolutionary novelty? So historically, there have been kind of 3 criteria for novelty. So first, it has to be something new. So this can be a new trait, it can be a new behavior, or it can be new combinations of existing traits and behaviors. Second, this newness should occur within a single evolutionary lineage. And then lastly, it should allow the organism to perform a new ecological function. And these are some of my favorite examples of novelty. So here we can see the elongated rostrum of the sawfish, and that's an example of morphological novelty. Down here, you can see tool use in crows. That could be an example of behavioral novelty. And then finally, this is the Venus flytrap. I would say this is an example of combinations of a whole bunch of weird stuff. All right, so why is it important to study novelty if we want to understand diversity? So the simple answer is that novelty can be extremely generative. So what do we know? So first, we think that there are many origins or mechanisms that can produce novelty. So this could include genetic changes, changes to developmental processes, or changes in the environment. And we know that changes at any of these stages can lead to potentially infinite downstream effects. The second thing that we know is that novel traits such as mammalian teeth, pharyngeal jaws in fish, and then the toe pads of anolis lizards, they're not only tied to ecological divergence, but they're also tied to adaptive radiation. And in fact, these are often referred to as key innovations, meaning the evolution of these traits was necessary for organisms to first enter that niche. And then finally, evidence suggests that there should be a relationship between ecological divergence, reproductive isolation, and speciation. So for example, if ecological divergence results in a large geographic range, you can see allopatric speciation. Alternatively, we might see selection against hybrids, and that can result in an increase in isolation via reinforcement. And we might even see that natural selection for an adaptive ecological trait incidentally produces isolation if there is a shared or linked genetic basis to that trait, and that's often referred to as a magic trait. So despite what we know, there are still a lot of unknowns. So what are we missing? First, I think it's likely that, I've just told you it's likely that there are many different origins or mechanisms that can produce novelty. However, very few studies directly tie an origin to a specific novelty. And then potentially more importantly, very few studies investigate the origins or mechanisms in an evolutionary context. Second, we do not know how the evolution of novelty affects diversification along other trait axes or dimensions. And I want to explain this using the example of pharyngeal jaws. So pharyngeal jaws are key innovation for labrid fish, and they allow them access to this energy-rich predator niche, right? And we do see a lot of variation and diversification in the key innovation in that trait itself. However, we also observe shifts and variation in behaviors, so different feeding preferences and habitat preferences. And we also see new variation in other morphologies that are seemingly unrelated to this. So coloration, for example. So it's clear that the evolution of novelty has effects at an organismal level. And we don't understand, number one, all of the changes that occur, nor do we understand if and how they all fit together. Finally, we currently don't have a lot of empirical data that explicitly connects variation in novelty to the strength and direction of reproductive isolation. So addressing these gaps is necessary if we want to start predicting if, when, and how diversification will occur. So today I'd like to talk to you about my dissertation work in 3 distinct parts. And these parts are going to be in the exact opposite order of what I've explained, just because. So first I'm going to tell you about the relationship between novelty and reproductive isolation. Second, I'd like to talk to you about how novelty is highly dimensional. And then finally, I'd like to tell you about some of the insights that I've made regarding the genetic basis of novelty in the system. And before we jump into the research kind of stuff, I'd like to introduce you to my system. So I study novelty using the Pupfish system, and they are a great system for a few reasons. So first, I'm going to begin by showing you the generalist pupfish. So here they are right here. Woo, there we go. This pupfish species is found across the Atlantic coast of North America, throughout the Caribbean, and even into South America. The generalist pupfish has a generalist diet, so they eat algae, insects, and detritus. However, if we zoom into San Salvador Island in the Bahamas, we actually find a young adaptive radiation of pupfish, and this includes 2 specialist species, the snail eater and the scale eater pupfish. So adaptive radiations are characterized by diversification across ecological niches, and this is the case in the pupfish system as well. So about 30% of the snail eater's diet is snails, and about 50% of the scale eater's diet is the scales of other pupfish. So aside from being very interesting and cool niches to explore and study, this variation in diet and ecological niche also makes pupfish a very powerful system for hypothesis testing, because we essentially have a gradient of ecological diversification and novelty, where we see generalists and snail eaters have quite a bit of niche overlap. Snail eaters and scale eaters have intermediate levels of overlap. And then finally, we see the most diversification between generalists and scale eaters. All right, so I'd like to show you the pupfish specialists in action. So both of these specialists are outstanding examples of evolutionary novelty. So first, the snail-eating pupfish exhibits a. Ooh, is that my mother?
Yeah.
It's my mother. Okay. Mom, can you mute yourself? Here. Okay, I got it. We're good. Okay. All right. So first, the snail-eating pupfish has an outstanding example of a novel morphology, and that is actually the extension. She unmuted herself. I'm linking, help me out here.
I don't know if I can. I don't have any control.
Okay, I muted her again. Carly, you have permission to meet my mother if she does. Okay.
She just made a crack with
How? Okay.
Turn your sound up. Yeah, you could do that.
Sorry, you're right. You guys are right. Okay. Somehow. Oh, shoot. Okay. Oh, okay. Hopefully it doesn't happen. We'll see. All right. Okay, we're back. Snail-eating pupfish have a novel morphology. We call it the novel nasal protrusion, and it's actually this bony extension of the maxilla. And we hypothesize that it is adaptive for oral shelling. And oral shelling is a novel behavior that the snail eater performs, where instead of crushing the shell, they actually wedge that snail out of the shell. She's gonna get a talking to later, that's for sure. Sorry, I'm having a hard time. Okay, I think I muted it just because we're like connected here. Okay. All right, so a novel morphology and a novel behavior of oral shelling. The second novelty that I like to talk to you about is scale eating. So scale eating is performed in less than 1% of all fish species. It's extremely rare. And a lot of people want to know the nutritional value of scales. I don't have an exact like measurement of that, but essentially it's like eating fingernails or hair. So it's a very, very strange and unique ecological niche. There are probably a lot of morphological adaptations for scale feeding. However, one of the most outstanding, I think, are the large jaws of the scale eater. So on average, they are twice as large as the jaws of other pupfish species. All right, so in addition to knowing about the ecological niches of our pupfish, we also have a good understanding of the phylogenetic relationship between these groups. And the main thing that I want to point out here is that scale eaters form a single group, so they look to be monophyletic. Snail eaters also generally group together, with the exception of the crescent pond generalists and snail eaters, so we know that there is ongoing gene flow there. And then the last thing I want to point out is this is actually one of our outgroups. We have a couple of them here, but all of the pupfish species that I've talked about across the Atlantic and throughout the Caribbean are actually also performing in that generalist niche. So we're assuming that the most common recent ancestor for the adaptive radiation also was performing in the generalist niche. Okay, so with that Pupfish Primer, let's get into some data. So I'm going to begin by talking about the relationship between novelty and reproductive isolation in the system. So my driving question for this project is pretty straightforward. And the question is there a relationship between novelty and isolation? And if so, what is it? So I started off with the hypothesis that there should be a positive relationship between novelty and isolation. And I basically went in this direction because theory predicts that the more divergent a niche is, that should be accompanied with increased reproductive isolation. And that could be because all of the accompanied adaptations to perform in that niche have incidentally affected isolation. Or it could also be if the niche is so divergent, there may be strong selection against hybrids and hybridization. So based on this theory and empirical data, and what I've told you about the pupfish, these are the predictions that I'm going in with, where generalists and snail eaters should have the lowest levels of isolation. Snail eaters and scale eaters will have intermediate levels, and then generalists and scale eaters should have high levels of reproductive isolation. So to test this hypothesis, I measured isolation and isolating barriers in several different ways, and I'm going to show the results of 3 of them today. So the first way that I measured it is by doing sequential choice assays for mate preference in the lab, and I used the number of eggs produced from a pair as a proxy for preference. And I really just like showing this video of me hand-picking eggs. It takes forever. The second way that I measured isolation is by monitoring offspring viability across 3 developmental time points in the lab. And really here I was looking for differential survival between hybrids and purebred offspring. And then finally, I leveraged existing data in our lab where we have wild survival of a bunch of different hybrid offspring. And essentially I compared hybrids with phenotypes who look like parental types, like purebreds, to hybrids that were a true intermediate form. And zooming out, this means I have estimates of both prezygotic and postzygotic barriers and intrinsic and extrinsic barriers. So there are a lot of measurements and data that go along with this, but today I'm going to show you the cumulative strength of reproductive isolation as probability of isolation between groups. All right, so this is what this data looks like. On the x-axis, we have ecological niche differentiation, which is essentially a proxy for novelty. And on the y-axis, we have probability of isolation. So the first thing that I'd like to point out is that when we consider only generalists and snail eaters, they seem to fit my predictions. So we can see a generalist to a snail eater here and a snail eater to a generalist. However, generalist females and snail eater females actually have opposite relationships with the scale eaters here, where generalists have lower levels of isolation compared to what we would predict based on their niche. And snail eaters have extremely high levels of isolation. The final thing I want to point out, which is the elephant in the room, which is the scale eater results over here, where there's essentially no isolation between either of those groups. So to put that into a biological context, this means that scale eating females actually preferred to mate with heterospecifics. The hybrid offspring in the lab had higher survival, and then in the wild, the intermediates are actually doing better than the parental types. So my conclusion from this study, first and foremost, is that the relationship between novelty and isolation is not linear. So I propose that the linearity of that relationship may depend on the complexity of novelty being examined. So complex novelties make it more difficult to predict levels of isolation. Furthermore, I think that this study shows that many of the common barriers, or at least commonly measured barriers to gene flow, are still unformed in the system. And this might suggest that we need to look at other isolating barriers, so temporal isolation, habitat isolation, and then fitness of hybrids in parental niches. I'm like, can you kick her out? Okay, so next I would like to talk to you about how novelty is highly dimensional. But my punchline here is going to be that novelty may primarily arise on the behavioral axis. All right, so my guiding question for this part of my thesis is essentially, what are the additional changes that occur when novelty evolves? And if and how are they adaptive to whatever novelty we're examining? So the 3 dimensions that I've explored during my PhD work are the dimension of morphology, behavior, and then kinematic traits. And I'm going to talk to you about each of these. So first, there's good evidence to expect shifts in morphology to accompany evolutionary novelty. So this is a classic school of thought that form is tied to function. I also think it's supported by the fact that we know that some key innovations are necessary. So some morphologies are necessary before entering into an ecological niche. So I explored this in the pupfish system, and I use the snail eater explicitly here. And naturally, from everything I've told you, my question is going to be if this nasal protrusion is adaptive for the behavior of oral shelling. And my prediction is that if it is adaptive, fish with larger nasal protrusion should be able to eat more snails. All right. So before I get into exactly how we did this, I want to acknowledge a wonderful undergraduate researcher, Christy Dixon. I think I saw Christy here. So Christy was one of the first undergraduates that I got to work with during my PhD. And I honestly credit her for like the reason why I enjoy doing research with undergraduate students. So Christy was one of the smartest, most inventive and students that I got to work with, and she was honestly just a joy to be around. And a lot of this work is due to all the effort that Christy put in. All right, so to test the prediction that the nasal protrusion size is related to oral shelling, we gathered our 6 focal groups. So we have the 3 species from San Salvador Island. We measure performance in a generalist outgroup along with F1 and F2 snail-eating hybrids. We put these fish through performance assays where we gave them 5 snails in 48 hours to consume them. And then we came back, we could see what proportion of snails were eaten. All right, so what we found is as follows. So first I'll point you to the nasal protrusion. I keep forgetting I'm on Zoom as well. Okay, so we see in this graph the results of nasal protrusion size across groups. And the things I'd like to point out is that generalists and F1 hybrids have smaller nasal protrusions than the purebred and F2 hybrids, right? So we can see this variation in difference in morphology. However, we have no difference in snail eating performance. So explicitly, you can see that the snail eaters and their hybrids perform at similar levels. And even the generalist on San Salvador Island is consuming statistically similar numbers of snails. So in total, we found that nasal protrusion size does not seem to be adaptive for eating more snails. However, this could be due to the experimental setup. So it still might be adaptive for things like locating and finding snails or in reducing handling time. Second, this data supports that oral shelling may be primarily a behavioral adaptation. And I think a good piece of evidence for that here is that our generalists on San Salvador Island, who we know are experiencing gene flow with the snail eater, are actually consuming more snails than outgroup generalists. And this is despite the fact that they have, a non-existent nose, basically. And then finally, I think that this study opens up the possibility that that novel nasal protrusion might be used for something else. So something like species recognition or mate choice. Okay, so that was the relationship between novelty and morphology. Now I'm going to talk to you about the relationship between novelty and behavior. So I think there's also good evidence to expect that behavioral shifts might accompany the evolution of novelty. The behavior first hypothesis particularly or specifically predicts that plastic shifts in behavior may precede other adaptive changes, specifically morphological changes. And then I also want to point out that previous empirical and theoretical work on like this aggression boldness continuum also predicts that increased aggression and increased boldness would be useful for entering a new niche. All right, and I have a little piece of circumstantial evidence to kind of convince you that specialists should be more aggressive. So this is a generalist just picking away at plants, essentially. And then this is a video of this I took with my iPhone in literally a plastic bag in the Bahamas. I'll play it a couple times, but the scale eater just like leaves a puff of smoke where the prey item was. Right here. So some circumstantial evidence that I think these specialists might be more aggressive. So to investigate this, I measured aggression across 5 different groups. So our 3 species on San Salvador Island and 2 generalist outgroups. I did this using mirror assays in the lab, and this is what this looks like. So you show a fish their mirror image, and then they get really angry and attack themselves. And we use the number of attacks performed during an assay as a proxy for aggression. All right, so here are some results. On the x-axis, we have our different groups that were tested. On the y-axis, we have the number of attacks that were performed during an assay. And the first thing I want to point out is that we did indeed find increased aggression in our specialist species, particularly when we compare them to the outgroup generalists. I also want to acknowledge that we had a significant effect of an interaction between species and sex. So this kind of means that the pattern may have been driven by increased aggression in scale eater males and also some increased aggression in snail eater females. And then I just wanted to point out a fun kind of part of this data, which is that our scale eating females never attacked in these assays, which I think is really interesting. So one of my pet hypotheses here is maybe these females have increased ability to recognize conspecifics to prevent aggressive encounters. All right, so we've tackled morphology and behavior. I also looked at or explored whether shifts in kinematic traits accompanied evolutionary novelty. And I did this by specifically focusing on the scale feeding in the system. So my hypothesis is that novelty likely requires changes in feeding kinematics in order to occupy a new ecological niche. And this is because changes in any kind of trait might affect feeding kinematics. So the analogy that I wanted to use to kind of explain this thinking, or at least put it out there into the world, is a baseball player's swing. So if these are the kinematic traits that we are measuring to get a home run, if that baseball player changes their stance, so it's a behavioral change, these kinematics are they no longer fit and will need to change if that baseball player still wants to hit a home run. Similarly, if the stance did not change, but their body was a different shape, those same initial kinematics would no longer fit and they would have to be changed, right? So again, all of this is to say that I think scale eating kinematics will be quite different. All right, so to measure feeding kinematics, I took high-speed video of generalists, scale eaters, snail eaters, and then F1 hybrids performing strikes on scales and shrimp. And this is what that video looks like. Good job. So from these videos, I was then able to measure a suite of kinematic traits that we thought were particularly related to biting, right? And this is what we found. So first, for the results of this LDA, you can see that scale eaters cluster over here by themselves. There's my cursor, here we go. So scale eaters cluster by themselves and are indeed divergent. So these are, the LDA contains all of the measurements that were taken from these videos. And if we look at specific measurements, we can see that the lower jaw angle of scale eaters is actually higher, meaning it's more obtuse. And we can also see that peak gapes, average peak gape sizes, are much larger for scale eaters. So these patterns are informative and they provide support for that shift in kinematic traits. However, they don't directly test or kind of answer whether these shifts are adaptive for scale eating. So to test this, we filmed fish striking these uniform cubes. I'll show you a video. And I want to say I made every single one of these cubes by hand. It was very labor intensive. So we would film them taking bites out of a cube. They'd take one bite, I would remove that cube, and then I can measure the area that was removed from that bite. And this was a very powerful data set because it meant that I have variation in kinematics tied to bite performance. And here I'm assuming that bigger bites are better. All right, so this is what we found. On the x-axis, we have peak gape size. On the y-axis, we have a proxy for bite size. So negative numbers are smaller bites, positive numbers are bigger bites. So the first thing that you can see is that as peak gape increases, so does the bite size. So this is essentially suggesting that the shift in peak gape is adaptive and does help with increasing that bite size. However, I want to take this inference a little bit. Oh, here I should mention, when we superimpose all of our means onto this, we can also see that the scale eater is the only species that can produce a peak gape large enough to actually get to that optimum, right? So now I want to take this a little bit further, which is I've already told you that the scale eaters have these massive jaws. So it shouldn't be super surprising to know that as their jaw size increases, so does their peak gape. However, their jaw size actually affords them gape sizes that are much larger than this 4.5 or 5 millimeter peak gape. So what I think is actually happening is that they are behaviorally mediating how wide they open their mouths during an attack. So we know that they're actually not depressing this jaw as far as they could. They could very easily have this 130-degree lower jaw angle. Instead, they're keeping that jaw flatter at 150 degrees, which in turn makes their peak gape smaller and keeps them on this performance peak so they don't overshoot it. So the main conclusion from this section is that scale eaters do have divergent feeding kinematics, but they are behaviorally mediated. And it's this combination that ultimately places them on a performance peak. Furthermore, I just briefly want to point out that I did measure the F1 hybrid performance, and they could never get up to where the scale eating specialists were performing. And so this might be kind of first evidence for a source of extrinsic isolation. Right, and then zooming out even further to put all three of these studies into the context of a relationship between novelty and dimensionality. I've shown you that we currently don't have evidence that morphological adaptations are there for snail eating. but we do see that the large jaws of scale eaters might be important for peak gape size. We currently do have evidence for behavioral shifts in aggression for both specialists. And then we also see that scale eating performance is not reliant just on that morphology or just that behavior, but this unique relationship between the two. So I think that together, this may be seen as support for the behavior first hypothesis. So we always saw behavioral shifts in both of those specialists. I think this also might suggest that more complex forms of novelty might require adaptations across many more dimensions. All right, finally, I'd like to talk to you about how I've begun to explore the genetic basis of novelty in my system, and explicitly in an evolutionary context. So identifying mechanisms and origins for novelty can be very important and informative because it can help us make predictions about when novelty may evolve, and it can also help us make predictions about any of the downstream effects that might occur. All right. Before I jump into this section, I want to talk about another undergraduate researcher who is sitting here today. We can all stare at her, Julia. So yay! Julia has helped immensely on this project, and I have a section dedicated to you later on. So for now, I'm going to say that they were absolutely integral to this work. Okay, so the hypothesis here is that essentially novelty might require new mutations. And I'm keeping that as a very simple hypothesis and prediction going forward. However, I do want to acknowledge that I just got done telling you about how complex these traits are in the system. And then we also know that the radiation is extremely young. So it's estimated to be 10 to 20,000 years old. So I'm mentioning this to say that it might be unlikely or it might be unrealistic for me to expect that all of our novel traits are going to be underlined by de novo mutations. So in addition to looking for increases in de novo mutations, we could also look at standing genetic variation being used and introgressed variation being used. All right, so to understand how novel traits on San Salvador Island were brought about via de novo mutations, we first needed to identify regions that were associated with these specialist traits. And to do that, I took a fairly traditional QTL approach with some caveats. So some unique aspects of this study that I'd like to highlight are first, I actually produced F2, or I did, Chris did, produced F2 hybrids from two separate ponds. So this is Crescent Pond and Little Lake. So these ponds are separated by 6 miles on San Salvador Island, which is a long way for a fish to walk. And they also have quite high FST between the two. So from like a methodological standpoint, I'm using these as independent samples. The second thing that I want to mention or explicitly mention is that these crosses that we produced, or the F2s that were produced from the crosses, were made explicitly from scale eater and snail eater parents or grandparents. So the phenotypes that we're looking at or identifying here are ones that are going to be related to the specialist niches, right? Okay, so samples from Crescent Pond and Little Lake, we phenotyped 30 craniofacial traits along with an additional behavioral trait. And then we want to connect our genetic variation to that phenotypic variation that I just talked about. So we sequenced these using RADseq. I aligned the data to the pupfish genome. We called variants in GAT-K. I estimated linkage maps and then scanned for significant QTL regions. I want to highlight that I did these independently for both ponds. So I essentially made twice as much work for myself. After identifying a region, a significant QTL region, this is where I got kind of really excited. Because then I could ask these questions that I think kind of really become intellectually interesting. So number one, what's in that region? Number two, what does that region do in the other lake? Number three, is this region important in the wild? And then finally, do we see an increase of de novo mutations within these QTL regions? So I'm going to kind of walk you through that question pipeline using an example here. So first, I independently detected significant QTLs associated with cranial height in both Little Lake and Crescent Pond. And reassuringly, they map to the same place in the pupfish genome. So that's great news. We were then able to look at the regions that overlap between those two ponds and characterize the genes that fell within that region. So we found that 44 genes were near or within the overlapping region between our two QTLs. Next, I leveraged the findings of my brilliant lab mate, Emily Richards, to search within these 44 genes for known adaptive alleles. And here adaptive alleles mean that they are alternatively fixed between specialists and under a hard selective sweep in the wild. This narrowed that list of 44 genes down to a list of just 3 genes. And I want to point out that one of those genes, GNAC, has popped up in a couple of the studies that we've done in the lab. And we know that it's associated with aggression and microcephaly. So I followed that same methodology for all the significant QTL regions that I found, and I worked my way down to this list of genes with adaptive alleles, and then I can investigate how those adaptive alleles got to San Salvador Island. So I again leveraged Emily's wonderful data set here, where she had already identified the adaptive alleles across the entire genome that are characterized as de novo, standing, meaning that they can be found outside of the radiation, other places in the Caribbean, or if they're categorized as being introgressed into our pupfish system. And the power of combining this quantitative genetic and population genetic approach is that we now have specific phenotypes that are associated with this genomic region, which in turn are tied to adaptive alleles under selection in wild populations. So these are the patterns that we found. So first of all, a lot of the adaptive alleles within overlapping QTL regions were categorized as standing genetic variants that are found other places across the Caribbean. However, these proportions are actually less than what was expected by chance. And instead, we can see, so here are my predictions for, predictions for de novo and introgressed variants. Instead, we found an overrepresentation of introgressed adaptive alleles that fell within those QTL regions. I do want to say there are, of course, some de novo mutations that we found, but they fall within the expectations of, randomly distributed throughout the genome. So what have we learned about the genetic origins of novelty? So first, we found little evidence to suggest that de novo mutations are used disproportionately for phenotypes related to novelty. And instead, we saw this overrepresentation of introgress variation. So introgression and hybridization may have been very important for the origins of novelty in the system. I think the natural next questions are, why are we only seeing novelty on San Salvador Island and nowhere else if these alleles are other places? And some things that I can think of would be, there could be a unique G/E interaction that is specific to San Salvador Island, or it might be that we needed this combination of those introgressed alleles and we aren't seeing that other places. Okay, so in conclusion, I started this talk by pointing out some gaps in our knowledge, and I'd like to put forward that I think my thesis has begun to address some of these. So first, for that QTL study, it suggests that novelty doesn't necessarily mean new, at least at that mechanistic or origin level. And instead, that introgressed variation might be exceedingly important for the origins of novelty. Second, my chapters exploring the multivariate nature of novelty strongly suggests that shifts along that behavioral axis are probably at least important in the initial stages of novelty here. And then finally, my last chapter, whatever order you want to say, this reproductive isolation chapter, I think, has contributed one of the few empirical estimates and connections between novelty and isolation between species. I think what I found is that our current theoretical or current knowledge actually doesn't predict what we're seeing in the wild. So we're clearly missing some piece of the puzzle. I'm gonna drink a water. So as Chris so nicely set me up for, I've done a lot more in my PhD than perform research. So I'd like to share some of those accomplishments with you today. So first, I was able to participate in the SurfSmart program here at Berkeley. If you haven't heard of SurfSmart, it's a wonderful initiative that pairs up undergraduate and graduate researchers to perform science over the summer, and it's an absolutely wonderful experience. But this is the program where I was paired up with Julia, and I truly couldn't have asked for a more insightful, talented, and kind person to work with. So Julia, you are obviously very integral to the completion of that QTL, like, thank you so much. And Julia is actually currently working on their own honors thesis. And they're taking a long time to pick a date and tell us when it is, but you're all invited, and you should come and hear all the really cool work that Julia is doing on looking at how correlations between traits either constrain or aid in diversification. Second, I was able to participate as a graduate student research fellow, whatever the title would be, for the REU program here at Berkeley. So working with this program allowed me to accompany 10 amazing undergraduate researchers on their kind of first steps into research. And getting to hear about their progress each week and seeing them grow and succeed in science was one of the most joyful experiences I was able to have during my PhD. And I was able to do this last summer, and I gotta say, like, having this experience with new researchers at the end of your PhD was especially like joyful. It gives me a little lift to be like, look at how great science is, especially when you're trying to finish. And then lastly, one of the accomplishments I'm most proud of is that Emily and I were able to co-develop and teach a course called Seeing Yourself in Science. And we designed this course to essentially offer a safe place for undergrads to just like get experience with research where there wasn't this pressure of understanding biological concepts or things like that. So in our course, students were able to collect data and answer questions on anything that they could come up with. And one of the really great observations is that students actually usually pick topics that are directly related to improving some aspect of their life. So during this course, we would lead them through the scientific process. So data collection, data management. We taught everybody to code and analyze things in R. They gave a final presentation. And I'm very proud because we wanted to explicitly have this class be a confidence booster. And from our initial look at our pre and post surveys from the course, we can see that we saw marked increases in networking, which, I don't know why we called it that, but essentially, like, how comfortable do you feel talking about science with other people? And also in science identity. And one of our questions is literally like, do you feel like a scientist? So being able to see the increase is really great. Okay. Okay. I have this printed out. I'm going to read it because I'm fairly certain I'm about to cry. So we're going to go with this. So the first person that I would like to acknowledge is myself. Yay! So I want to start off with this because I don't think we give ourselves enough credit for the work that we do in pursuing research. So I'm officially going to say in public that I'm very proud. I have 6 pages. Very proud of the research and accomplishments. And I'm also proud of all of you for pursuing your own research. Oh. Next, I'd like to acknowledge my family. So the support, care, and love that I've received from my family leading up to and during the process of getting the PhD is invaluable. Each of my family members, including my grandparents, parents, my brother, and my in-laws, have provided me with outstanding emotional support during the process. And I want to acknowledge that having a family that you know you can fall back on has made this process significantly less scary. I'd like to thank each of them for letting me talk about fish for so long. I don't anticipate that decreasing anytime soon, but here's a public thank you. Next, I'd like to acknowledge my furry family members, including my 2 cats, Tulip and Alfalfa, my dog, Tomatillo, and my family dog, Mamie. I don't think you were watching this talk, but I think many of our first experiences appreciating animals comes from our pets, and I'm no exception. So thank you for being a continued source of joy in my life. Next, I'd like to acknowledge and thank my husband, Matthew, for moving not just once for my PhD, but twice. Matthew has also helped me with many tasks directly related to my PhD, such as fish care, field work, and proofreading drafts. But the thing that I'm most thankful for is that I never felt that I was doing this alone. Moving and pursuing a PhD was very scary, but knowing that you were always there for me made it feel like it was easy to handle. So thanks for making me a braver and stronger person. And I hope you're ready for move number 3. So next, I'd like to acknowledge and express my gratitude for my research experiences before pursuing a PhD. So specifically, I'd like to thank everyone who was present and supportive during my master's degree. So first, I'd like to start by acknowledging my previous advisor, Becky Fuller. So Becky thank you for teaching me about fish. Thank you for teaching me to code in R. And most importantly, thank you for making me feel like I could be a successful scientist. I actually don't think that I would be pursuing academia and research as a career if it wasn't for my experiences with you. Second, I'd like to thank my lab mates for my master's degree, so Liza and Rachel. Thank you for being exceptional lab mates and friends. You showed me how collaborative and supportive the scientific community can be, and I'm a better scientist having developed alongside you. I'd like to also thank everybody from that department in general. So Sam, Laura, Aaron, Sarah, Miles, and Ree. That community that you all provided sincerely shaped who I was as a person, or am as a person and scientist, and I'm very thankful for your continued support even to this day. Okay, so for those of you who don't know, I actually started my PhD at a different school at the University of North Carolina. So I'd like to acknowledge the support and acceptance from that community. So specifically, I'd like to say thank you to Kate, Kamal, Megan, Catherine, Kayla, and Amy. So you're all amazing scientists and friends, and I'm very thankful that I got to start this journey with you all. Okay, I'd like to acknowledge the IB and MBZ communities for welcoming me here, essentially. So moving in the middle of the PhD, again, was very scary. The inclusion and acceptance over the past several years has been integral to my success as a scientist and as a person. I could literally go around and name every single person in this room, so thank you all very much. Specifically, I'd like to say thank you to Sylvia, Jackie, and Daisy for making me feel accepted in that first year, despite the fact that I was very clearly a grouchy old grad student. And then I'd also like to say thank you to Mallory and Molly. So thank you two for accepting me as the grouchy old grad student right off the bat. And thank you for always talking science with me and for being great friends. All right, almost done, I promise. I'd also like to thank all the members of the animal behavior group. I see a lot of you here. So this is probably my favorite class that I've taken at Berkeley, and maybe my favorite class that I've taken ever. So your feedback has made my science better. Your kindness and humor has made me a better human, and I'm extremely thankful to know that that community is thriving. I'd also like to take this time to thank Eileen and Damien. So you're two of my committee members, but in addition to that, I'd also like to thank you here because you're the heads of that animal behavior group, and I truly think that that community is as supportive, kind, and insightful as it is because of your outstanding leadership. So you've not only fostered a great community, but you've also both provided me with invaluable feedback on and recommendations regarding my academic work and progress, and I'm very thankful that you are on my committee. All right, to my current and past lab mates, including Joe McGurr, Joe Harris, Austin, Fania, and David. Thank you for being there with me. Having caring, knowledgeable, and fun lab mates has made this experience infinitely better. I've learned so much from each of you, and I'm excited to work with you all in the future. That's right, you can't escape me just because I'm graduating. I'd also like to acknowledge all the undergraduate researchers that I've worked with over the years, especially Christy Dixon and Julia Dunker. I'm truly a better scientist and mentor having worked with you. Okay. Chris, and I wish I could zoom in here. Let's see if I can. Oh, wait a second. It's a highlight.
That's me in the mud.
Yeah, this is Chris in the mud. That's right. Okay. So I'd like to acknowledge and thank my advisor, Chris Martin. So thank you for #1, deciding to do research on the pupfish. I really enjoy the system. I'm very grateful to have had the opportunity to work with them. Thank you for accepting me into your lab. I'm not sure if you saw this as a risky move, but having someone give you the opportunity to pursue research is a big deal. So thank you for giving me a shot. Thank you for taking me to the Bahamas. And today I got a notification from Google that that was actually 4 years ago, the last time we went. But getting to do field work was a definite highlight of my PhD, and I hope that we can make it back soon. Thank you for taking me to Berkeley. Thank you for also all the financial support you've given during the entire PhD process. And then lastly, I'd like to say thank you for helping me to become an independent and inventive scientist and an overall expansive thinker. The lab environment that you've created is truly integrative. I appreciate how much knowledge I've gained by simply being a member. So I'll say the same thing that I said previously to the lab members, which is I look forward to working with you in the future because you're never getting rid of me. All right, and then lastly, I'd like to acknowledge I'm gonna cry. My best friend. Okay, give me a second. My best friend and lab mate, Emily Richards. So it's extremely difficult to express how large of an impact you've had on me as a scientist and as a friend. So I've told this story before, but one of the biggest reasons that I actually joined Chris's lab is because Emily was my host. And she was such a kind, funny, and personal student that it made me confident that if I went there, I would have at least one friend. So Emily has influenced me immensely scientifically. And I think a lot of that was evident in portions of my talk today. But having a friend who is equally as excited and inquisitive about the same research topics as yourself is truly transformative as you're forming research ideas and carrying out experiments. Brainstorming sessions, discussions, and research with you has been one of the largest and most important influences I've had during the PhD. I'm extremely grateful to have had you as a lab mate. Furthermore, Emily, you're one of the kindest and most empathetic and understanding people that I know. And having you alongside me during this process is frankly probably one of the only reasons that I made it through. So thank you, Emily, for being a great friend and a great scientist.
And you're never getting rid of me.
Great. Okay, I'll pull myself together. But with that, I'm happy to accept any questions about any part of this. That's Michael.
So that was spectacular. It was just a ton of super interesting work. I was confused about the QTL stuff.
Yeah,
Did
me
you
too.
find the six QTL in both mapping populations?
Yeah, so okay, cut this information for time, and I'm happy to talk about this because this is still something that I'm wrestling with and I'm addressing in revisions. So for some traits, yes, I was able to detect, and cranial height was my nicest one choice, so I showed it to you. So it was the same exact region. For other QTL regions,
The same six?
sorry, it's the, so okay, for that, so that's a QTL number 1, and that's where we had the same trait mapping to the same exact location, right? That's 1. So then I had 6 more or 5 more to make total. So like traits number 2, I often found that correlated traits were mapping to the same exact location in the different populations, which is something that's really interesting to me. I think there are a couple options to why that might be. So it could be that that particular region is affecting multiple traits, right? And I think on average, one of the Stickleback studies says that a single locus can affect up like 8 traits. So it could just be this one region affects many correlated traits. I think it could also be that we're picking up based on our power in our studies, like if a trait is polygenic, like I'm just picking up different ones for different reasons, essentially. So some statistical power there. But I found 6 significant QTLs. 5 out of those 6 always had a corresponding correlated trait in the other pond. There was only 1 QTL that I found in one pond and not the other. Yeah.
And what was the effect size of those? big
Pretty
QTLs?
moderate, like 7%. You know, yeah, pretty moderate. Yes.
I was really curious about the snail nose
Me
thing.
too.
So in the system, you said, is the snail morph just on that island or it's also other places? Only on,
Only on the island. Yep.
okay. And so all the different populations in the phylogeny you showed,
So all on the island. Yeah.
those are all within the
Yep.
island.
Yep.
It's just so weird.
I
don't know
agree.
if you want to speculate more. I'm curious.
For just like what the nasal protrusion is used for.
Yeah, like
Kind of.
does it actually have to, do all snail eaters have that? Like potentially are you missing some because you're biased in who you're reviewing?
So for right now, I'd say like all of the samples of snail eaters definitely have noses. Let me go back to, where did my cursor go? It just keeps disappearing. Let's go back to, so you can see how terrible I am at this. There it is. Oh shoot. Okay, well, we're gonna give up on that. But I think in general, sorry, all of those snail eaters have noses. The generalists also have, like, I think we can measure a small nose, but if you had noticed when I was showing the nasal protrusion side stuff, scale eaters were not on there because they have absolutely no nose. I can't measure zeros over and over and over again, right? I think we do see some variations across the populations in nasal protrusion size, but as far as like proportion of the diet that we see in the wild populations, they're still pretty similar, right? So it doesn't seem like all of the kind of, so you know there's variation in these nasal protrusion sizes, maybe we'd see some corresponding dietary effect, but we don't see that.
And none of the generalists have the nose.
Like, so like very, very, very small. So like when you look at them, you're not like, you have a nose, right? It's just like the kind of
Okay.
top of their mouth. Yeah. Definitely very weird. It could be, so I could think it could be used for something like species recognition, potentially. It could be something that's, you know, just an incidental trait brought along by something else. But yeah, and I think we could also see effects on handling time potentially. Yeah.
Awesome talk, Michelle. So thank you. Just to go off of what Becca said a little bit, and I'm sorry if you mentioned this, but was there any dimorphism in the nose?
No.
size, or
No.
absolutely none at all.
No, yeah, and I think,
so interesting.
yeah, so I definitely looked at that. No sexual dimorphism. So I like, again, I thought the same thing. Maybe it's a sexually selected trait. It does not appear to be so. However, I still think it could be like conspecific related. So at least you would say like, oh, this is all my same type potentially. But yeah,
That's crazy.
Kat.
So for the like mouth shape size and the height amount, like did you see any correlations between like age and like how well they're doing? Like I must have heard like sort of take it off, like are some of them just really bad at it, or is that like the juveniles? Like,
That's a good question. So we did not have like age. They were all adult fish. However, I think like it would be very interesting to see if like scale eaters need to learn and like get better at performing scaling bites. I think that'd be definitely really cool. The tapering off, again, that was very exciting to see because that kind of means that there's a performance peak. It's not just like it goes on forever, right? But an interesting thing about that data set is like we had individuals perform several times. And so like one strike instead of kinematics, that fish produces a great bite, but maybe the next time they just like, you know, you could trip and fall into the cube, and then those kinematics are not great for, right? So it seems like there's actually like individual level variation too for kinematic strikes. Yeah.
It was an amazing talk.
Thank you,
Michelle. So I have two questions. The first one is, do you know if there are difference in the stomach, intestine,
like morphology.
Between the three?
That's a good question. So that would be a question for our previous postdoc, Joe Harris. He did look at that, and he also looked at the microbiome. And I think right now we don't have any real like smoking gun differences that would allow. So I think, are you thinking about scale eaters particularly? Like how are they digesting it? Yeah, I don't think we have like a smoking gun that would suggest there's like this one adaptation. But yeah, I think their stomachs look pretty similar.
Yeah, and the second is some of the SNPs that you find, of the genes that you find, have you seen reported for kind of same diet in another species, in another groups? Parallel evolution.
Right. So again, so not being a model organism, so the annotations that we have to go on are often like mice or things like that. And so like we definitely do see associations, again, like the GNAC, we haven't tested that in our system, although I think Fania, wherever she is, there you go, we might be looking into stuff like that. And so yeah, like we do absolutely see associations with either skeletal traits or even aggression and things like that. So they do seem to like make sense, but I think again, with the caveat that we're not a model organism and we have to test it in our own system. Yeah. So yeah.
that was great.
Thanks.
I have a question regarding like how the scale leaders are maintained given their like weird mating preferences. So I think it was that like that the females don't prefer scale leaders over the others. So do you know if it's something with like male success is just really high, or if that's something that you're interested in looking at how that's maintained?
Yeah, I'm very interested in that. I think one of the things, again, if I could, let's see if I can bring this slide back up. One of the things that has stuck out to me in this data set, and I'm still working through this right now, but we actually have, that is not what I meant. Let me show you. Okay, so snail eater female to scale eater males, the isolation is really, really high. But this cross is also represented here with scale eater females to snail eater males, right? Like that is species level, the same cross. So I do think there's evidence right now for some sort of asymmetry and isolation. So like, this is even more complex than I initially thought. Something that I will say, like Emily had pointed out to me, another kind of interesting tying this all together is that scale eater females, again, were the ones who were like not aggressive at all in those trials. And so it's interesting to see like a scale eater female who's not aggressive is very accessible at heterospecific mating, and their hybrids do great. So there's a lot of complexity there. Yeah.
Sorry if I missed it, but did you do any like aggression experiments interspecifically?
So aggression experiments were always against their mirror image. So they would only be, they're seeing literally themselves. So they're always conspecifics, right? And it's also really important for fish because there's a lot of hierarchy with body size, right? So you can attempt to size match fish when you're measuring aggression, but like the best size match is essentially like a mirror image. So they're all conspecifics, yeah. But I mean, that is one of the outstanding like questions that haunts me here is like, are scale eaters indiscriminately aggressive or are they preferentially aggressive? Like, I don't know. Yeah, it's a great question.
Just
okay.
Do the scale leaders are trying to eat the scales from other scale leaders?
I don't know, but I forgot to mention that the pupfish are basically the only fish in these ponds, right? So scale eaters are literally eating the scales of only other pupfish. It's pretty wild. But yeah, I don't know if they will attack another scale eater. That seems more risky than attacking like a snail eater, but I don't know. Yeah, Dan.
When the scale eaters attack the other pupfish, does that cause mortality or is it kind of just a nuisance?
No, it's pretty much a nuisance, so they'll go away. I think we still have questions about whether like if you are left with like a wound or something, if that might, you know, have downstream effects, but it seems like they just kind of get attacked and move on. Jackie.
What's been your favorite part about teaching the Seeing Yourself in Science course?
Oh my gosh. My favorite part about teaching the Seeing Yourself in Science course, I think, has been getting to interact with students who are not primarily in biology. So I think another kind of aspect of that course is we want to target students who are in other departments. And so we had people in economics, we have a student in physics this semester. And so it's interesting to see like both the unifying questions that everybody's coming up with. So everybody wants to kind of answer questions about themselves in some way, but also just getting to interact again with like the Berkeley community at large outside of IB. It's been really fun. Yeah, Becca.
Do you see selection for like some difference in the scales? If they're all eating each, you know, if there's a lot just of their scales being eaten, is the mucus different or?
Good question. As far as I can answer today, the scales are not like, I cannot differentiate. It would be very useful if I could in the guts, especially to be like, oh, that's a general scale and that's a snail eater scale. So like, as far as just scale morphology, not really. I don't know about the mucus. That's a good question. And really a question of like, are the scales the actual target or is it the mucus membrane of some sort? Yeah. Yeah, Jim.
Have you already assessed, maybe Emily has, how much gene flow there is between these three phenotypes within, I mean, they're species, right? Within these lakes, is there?
Why would you attack me like that? Are they species? I don't know. Yeah, gene flow. So we do know, I mean, Emily's one with the numbers, but I do know like gene flow between snail eaters and generalists is the highest, right? And gene flow between generalists and scale eaters is much lower. Yeah. And then snail eater to scale eater, the lowest? Yeah, the lowest. Yeah, absolutely.
Does that match this?
No, it doesn't. So like snail eater to scale eater, the lowest that we just said matches this, but it certainly does not match this. Yeah. So I think, so again, just remember that, especially for this slide, this is mate preference in the lab, offspring viability across developmental time points, and then some brief measures of wild survival, but that was only for like 6 months. So we're missing any isolation before actually mating, and we're missing isolation past like 6 months of life in the wild. So like performance if you're a hybrid performing in a scale eater niche, like my kinematics chapter would say that you're pretty bad at it. So like that could be really strong and like, you know, helps with the isolation.
Do you see a lot of F2s in the natural population?
I don't know. That's a good question. That is a good question. Yeah, I don't know. Yes.
Nice lot.
Thank you.
And, the scale eating, so if there's questions about it, its nutritional value, could it just be a form of aggression?
I think, so okay, so actually a hypothesized origin for scale feeding is a behavioral shift in aggression where like this, the hypothesis goes that, you know, fish are fighting and then you incidentally ingest scales one day and now you're associating aggression with food, right? So that is definitely a hypothesis. I think that it could have maybe been, I think in some sort of way, a behavioral origin, right? But we do see increases in aggression for both snail eaters and scale eaters. So I think an argument for only aggression driving scale feeding maybe doesn't quite fit. And then I think circumstantial evidence that I would say is like we do see aggression for all these fish species. The males have territories, they're very aggressive. But scale eaters, like you can follow a scale eater across the entire pond and they're just attacking and attacking and attacking. And it's just like, it seems, again, we're totally anthropomorphizing, but it seems very intentional. It's not just like the behavior of defending a territory. Yeah. All right, I kept everybody here way too long. Cool. All right. Thank you.